Hey guys, what's up? So today in our AS biology lecture we're going to cover ribosomes in detail. So we need to understand at first what ribosomes are. So ribosomes, whenever we look at ribosomes, so first thing we need to understand that ribosomes can only be seen with an electron microscope. This is because ribosomes, they are very small structure inside the cell. They're very small, like kind of like spherical or round shaped structure which are located inside the cell, and these are actually not visible with the help of light microscope. Okay, we have talked about this is because light microscope's resolution is not enough because of the wavelength of light is 400 nanometers, and you can just see 200 nanometer resolution, but that's not actually enough to see this small ribosome structure. So we require an electron microscope to actually see these ribosomes in the cell. Okay, so now we need to understand that the difference in ribosomes in eukaryotic and prokaryotic cells. So 70S, there are two types of ribosomes, 70S and 80S. So at first, what does this S stand for? This S actually stands for Wedberg unit. Wedberg unit. Okay, so what does this Wedberg unit actually mean? So at first, let's understand that the 70S ribosomes are the ones which are usually found in the prokaryotic cells, and 80S ribosomes are found in eukaryotic cells. So this S stands for Wedberg unit. So this Wedberg unit actually means that the time which is taken for the sediment to be formed at a very high speed centrifuge. Okay, so at a very high speed centrifuge, the time which is taken for sediments to be formed at a very high speed centrifuge is what S actually stand for, and this is actually Wedberg unit. And based on this formation of these sediments, we actually denote these S's. So we need to understand this S does not actually, this is not a mathematical calculation. For example, 50S plus 30S is equivalent to 70S. This is not just adding 50 plus 30, because that would be 80, so that's mathematically wrong. So it actually refers to how much sediment is formed. So when these two ribosomes are enclosed together or joined together during protein synthesis, these 70S together, these two subunits actually make up a 70S. When they join together, they form 70S. So this is actually the Wedberg unit, which means the time or how rapidly substances can sediment at a high speed centrifuge. Okay, that's just additional information. We are usually not asked about this, but still, just to know, just to clear out this confusion. Sometimes some people does get confused. So 60 plus 40, for example, is 100, but it's not 80. But together when they join, this Wedberg unit actually comes 80, and this is not a mathematical thing once again. Okay, so now let's talk about the ribosomes. So this is a 70S ribosome. So 70S ribosomes are seen in prokaryotic cells, and 80S ribosomes are found in eukaryotic cells. Okay, so now if we talk about the structure of ribosomes, so what ribosome is made up of, so ribosome is actually made up of rRNA. This R actually stands for ribosomal RNA. So ribosomes are actually synthesized in the nucleolus of the nuclei. So it mainly consists of rRNA and some amount of protein. So mainly all organelles are actually proteins. Okay, so yeah, so main function of ribosomes, as I said, is actually protein synthesis. And ribosomes, there are two types of ribosomes, 70S and 80S. 70S is found in prokaryotic cells and 80S in eukaryotic cells. But there is also an exception to this. So in eukaryotic cells also we have two organelles, which is the mitochondria and the chloroplast. So chloroplast is obviously present in plant cells only, not animal cells, but plant is also eukaryotic. So these two structure actually has 70S ribosomes. So we are going to talk about why they have 70S ribosomes in detail in another video, where we are going to cover endosymbiont theory. Okay, where we are going to look at the evolutionary background of these organelles and how they origin from bacteria, which bacteria on the other hand is prokaryotic, which actually had 70s ribosomes, and over time, during evolution, these organelles were present within these bacteria, such as cyanobacteria had chloroplasts, and some bacteria required flagella to move, so they had mitochondria to release energy to provide the movement, and mitochondria is required for respiration, aerobic respiration, as we know. So yeah, so we're going to talk about those in a bit more detail in the endosymbiont theory. That's not directly part of the syllabus, but they do ask in certain questions that these two actually have 70s ribosomes, so we kind of have to know this thing. Okay, back to ribosomes. So back to ribosomes. So they are actually involved in protein synthesis, as I've already mentioned. So in protein synthesis, during protein synthesis, they actually allow different types of things such as mRNA, tRNA, amino acids, and certain regulatory proteins to actually sort of gather together. So these all substances can gather together at one place during protein synthesis. Gather during protein synthesis. So these ribosomes actually allow these substances to gather together during protein synthesis. So we're going to talk in detail about protein synthesis in the central dogma of biology in that chapter. So, and one more thing, the ribosomes, as we've already seen in our nucleus video, is that these ribosomes can be over here, they can be either attached to this rough endoplasmic reticulum, or they can also sometimes be found to be floating in the cytoplasm, in the cell cytoplasm. They can also be seen. So since that is an eukaryotic cell, that's obviously ATS ribosome. So these ribosomes can be seen floating around in the cytoplasm, or they can also be sometimes seen embedded in the structure of the rough endoplasmic reticulum. So yeah, that's it for this video. So what we covered is that these ribosomes, the 70s ribosome is found in prokaryotic cells, ATS is found in eukaryotic cell. This S actually stands for Wedberg unit, which is the amount of time which is required for substances to sediment at a very high speed centrifuge. So that's why when they add up together they form 70s, and this is not a mathematical thing. And then we learned about the main function of these ribosomes are actually in the protein synthesis, and certain eukaryotic organelles such as mitochondria and chloroplasts also have 70s ribosomes because of their prokaryotic origin, we can say. And we also say that certain substances such as mRNA, tRNA, amino acids, and regulatory proteins can all gather together during protein synthesis. These ribosomes actually allow them to clamp or gather together. So that's it for this video. We're going to talk about the other organelles in detail in the later video. Thanks for watching.